Miyano, how studying aspects of hibernation can get humans to Mars. And you can go whenever you're ready.
I want you to raise your hand if you would travel to Mars, if it was possible.
If it possible.
The journey to Mars may seem like the stuff of fantasy and fiction, but believe it or not, we may be closer to that idea than you think. When astronauts were sent on a long space journey in the movie Interstellar, they were able to do this by being put in a hibernation-like state. When they woke up, they were healthy and they hadn't had to endure the long journey. This could be the key to unlocking future space travel, and luckily, we have a few examples of this right here on Earth, animal hibernation. When winter is coming and there's no food around, animals will reduce their metabolism and body temperature to go into hibernation. This allows them to survive the winter without having to eat. Then, when spring arrives, they raise their metabolism and body temperature back to normal levels. They do this partially by shivering, but most of their heat is generated by a different and more flexible process. It's called non-shivering. Very creative name, right? Non-shivering is a hormone-driven process that burns fat to release heat. It increases metabolic rate, which we can then measure in the lab. My thesis project is investigating this process. So, to bring you back to my first question, if we want to bring humans to Mars, then we need to understand all aspects of non-shivering. My lab studies a mouse that is missing a portion of the biggest protein in our bodies, titin. Titin acts like a spring by storing energy during muscle movements. A previous study in our lab found that mice with the deletion of titin develop hypothermia at room temperatures, like the one we're sitting in now, because they shiver at a slower rate than they should. But what about when they're not shivering? I wanted to know if non-shivering was also contributing to the hypothermia of these mice, and if so, is it linked to titin? I used a hormone to maximally stimulate non-shivering in mice with and without the titin deletion. Then I measured metabolic rate, which allowed me to see how much heat they're producing. I compared the responses between mice with normal titin, shown in gray, and mice with a deletion of titin, shown in blue. As you can see by the responses in the graph, mice with a deletion of titin are not generating enough body heat, as shown by their lower metabolic rates. My results are the very first to show a link between titin, a muscle protein, and non-shivering. I want to bring you back, once again, to our human space travelers. If we can understand how titin regulates non-shivering, then this could be the key to bringing humans out of hibernation. More than likely, humans would need to activate non-shivering once they've reached their destination, whether that's Mars or another planet. My research can help pave the way towards understanding human hibernation and the potential for making distant space travel a reality.